Hello to my dear students. In our topic of the cell biology, that is the chapter Cell, the Unit of Life, in our last session we have discussed about the prokaryotic cell. Now in this session we will discuss about the eukaryotic cell. But before starting this session, let's talk about what do you mean by eukaryotic cell. Can I say this eukaryotic cell is present in eukaryotes, right? Now if I take the etymology means splitting of the term and getting the meaning out of it. If I see the etymology of eukaryotic cell, the U stands for the true and karyon stands for the nucleus. Means eukaryotic cell is that cell which is containing well-defined nucleus, means genetic material is enclosed inside the nuclear envelope. That type of cell you call it as eukaryotic cell, which is containing this eukaryon, or you can say eukaryotic nucleus. Now that is present in case of eukaryotes. What are the kingdoms which are included under the eukaryotes as per 5 kingdom system of classification? It will include the Protista, it will include the kingdom Fungi, even the kingdom Plantae, and even the kingdom Animalia. Fine. These kingdoms are belonging to eukaryotic kingdoms. But do remember, protistans are exclusively unicellular. Fungi will include few of that unicellular but mostly multicellular. In plants, the lower plants will be unicellular but higher will be again multicellular. Animals, you know that they are from the multicellular. But all comes under eukaryotes, which are having eukaryotic cell, which is having true type of the nucleus, means well defined nucleus will be there. Now in this eukaryotic cell, you will find there is a big variation between the plant cell and animal cell. We have discussed in the prokaryotic cell, they are having fundamentally same organization. But here we cannot say the organization is same, they will be differ. Like for example, if I take the difference between plant cell and the animal cell, the basic main differences, can I say in the plant cell, I am writing the features in the center of this line, then whether present or absent we will take it. Like the cell wall. Can I say cell wall is present in case of the plant cell but absent in case of animal cell. Now next, plastids, again it is present in case of plant cell but absent in case of animal cell. And if I take for the large central vacuole, that is a feature of mature plant cell, it is there. But if it is small, means if it is there in animal cell, it will be small, it will not be large central vacuole, which is singular one, it is not single. If it is there in animal cell, it will be small, many in number. So large central single is absent in case of animal cell. Next thing even to remember, next important difference, that is of centrioles. Centriole is very important difference which is going to be present in case of animal cell. Whereas in plant cell, it is almost absent in all plant cell. Right? So do remember, it's not clear while writing here. Let me to take this point slightly above so you can see and write it. I repeat, centriole under the plant cell, it is going to be absent in almost all plant cells. Now why I have written almost all plant cells? Reason, because the few lower plant cells will have the centrioles. So these are the main differences. Just now I have given you between plant cell and animal cell, which are the eukaryotic cell, means they are not same, they are going to be different. The eukaryotic cell is having compartmentalization, means their work is happening for individual organelle within their membranes. Very few will be membraneless. Like I have written centrioles, it will be membraneless cell organelle. But if I am taking ribosomes, even this is membraneless cell organelle present both in plant cell and animal cell. Right? Even ribosome, if you remember, we discussed in prokaryotes also. But there even it is membraneless. So can I say ribosomes will be the universal cell organelle which is present in prokaryotes as well as eukaryotes. Right? So ribosome can be the same thing which is there in plants in the case of prokaryotes and eukaryotes. But still difference is there. In prokaryotes it is 70S, in eukaryotes mainly it will be 80S. 70S can be there in eukaryotes in mitochondria and chloroplast, right? So with this small information, we have given the introduction to this eukaryotic cell. I again give the point that this is showing compartmentalization, means membrane bound cell organelle will be there. Even they are showing variety of complex. Even the eukaryotic cells will show variety of complex type of locomotory structures and even the cytoskeleton. These things are absent in case of the prokaryotes. In the prokaryotes, the locomotory structures are not much complex. They are going to be simple. Like for example, flagella. Flagella will be there in prokaryotes as well as eukaryotes. But eukaryotes flagella will be more complex as compared to the prokaryotes, right? Okay, now after this small introduction, let's talk about the cell membrane which is common in the prokaryotes as well as in eukaryotes. Their structure is similar in both the type of categories of prokaryotes and eukaryotes. So one thing let me to say, this session being very short, we are going to go with the important things. So please do focus on every aspect what we are taking, being we are selecting the important aspects which is important in your learning as well as to cover the NEET exam also, right? So I am taking first of all about the cell membranes. So cell membrane, can I say this is a delimiting membrane in case of animal cell. Why? Because there is no cell wall in case of animal cell. But in plant cell you will find cell wall, inner to that will be cell membrane. Now cell membrane, the composition if I will take, it is made up of lipoproteins. Lipoproteins means it is made up of lipids plus proteins, right? Their ratio, their percentage will varies from membrane to membrane. Like for example, if I am taking erythrocytes in human, their membrane if I will discuss, their membrane, then their lipids will be how much in percentage? 40%, and proteins will be 52%. Now if you take it a total, it comes out to be 92%. What about rest of the 8%? Right? What will be that rest of the 8%? That will include the carbohydrates which are oligosaccharides. Fine? You will find like even in your NCERT book there is a lipid bilayer on which the proteins are there. And you see in your NCERT book you will find the blue color portion which are lipid bilayer. I will show you on the slide next after drawing it, and embedded or the periphery the proteins are present. Let me to take the next slide to explain this part. There will be lipid bilayer with the head and tail. This is a one molecule of a lipid where this part you call it as head and this part you call it as a tail. Tail will be what thing? It will be hydrophobic, whereas head will be what? Polar, and it will be hydrophilic, right? Now if I will take second molecule opposite to that, being tails are hydrophobic, they will be facing towards each other and heads are towards outsides. of the cell membrane, right? So lipid is forming the bilayer, in which what is going to be embedded? The proteins are going to be embedded. If the proteins are embedded throughout from first upper layer to the lower, that you call it as completely embedded proteins, right? So what type of protein I have drawn here? This is an integral protein which is totally embedded or totally buried proteins, right? Some are partially embedded, like this one. If I will draw, this is even integral, but it is partially embedded. Fine, even with the covered by lipid bilayer. And you will find towards its outer side, if I will draw the protein which you call it as peripheral protein, they are not embedded inside the lipid bilayer. So can I say proteins are of two type here? One is the peripheral, second is what? Integral, which is based on ease of extraction. Ease of extraction means when you are studying on that, how much easy it is to remove. Obviously the peripheral protein is easy because it is toward outside. Integral protein, either it is embedded partially or totally. It is tough to remove out. So two type of protein is there based on the ease of extraction. And just now we have completed one of its composition, lipid and the protein. Where will be the carbohydrates? Carbohydrates will be forming the chains either attached to the proteins or attached to the lipids. If these sugars are in which form? That is in oligosaccharide forms. Then this protein, you call it as glycoprotein, and that lipid you call it as glycolipid, right? And protein, what you call it as? Glycoprotein. To which what is going to attach? The oligosaccharide, sugar part is going to attach. Just now I said, you know, RBC is having 8% of the oligosaccharide, which are the sugars. So they are attached toward outer side of the cell, not toward the inner side. Fine. And among the lipids, you will find here even the cholesterol, which is helping to give the fluidity. And the model, which is a modified model and most accepted given for this cell membrane, that is the fluid mosaic model, given in the year 1972 by which scientist? Singer and Nicholson, right? They had given that model where the fluid nature means quasi-fluid. The half fluidity will be shown, means it is not freely moving within the cell, it is not leaving its place, that you call it as quasi-fluid. This is shown by lipids, right, which makes the lateral movement of, it will show the lateral movement of the proteins. Like for example, see my fingers, these are, suppose these are the lipids, they are showing lateral movements due to quasi-fluid nature. If proteins are embedded there, can I say it helps in the movement of protein? So it is measured actually by the movement of protein, lateral movement of protein. Who is showing quasi-fluid nature? Lipids. And it is measured by the lateral movement of the proteins which are embedded in the lipids, right? So this is a quasi-fluid nature by the lipid. Mosaic pattern will be shown by proteins. Mosaic means like only patches are there of protein. Like suppose your room is there, na? The membrane of this wall, I am taking wall of the room like a cell membrane, it is in continuity. This is like a lipid bilayer. And in between you have doors and windows. What they are? They are like a proteins. So proteins are not continuous, they are in form of patches. That's why you call them as mosaic model, right? So who had given this modified and most accepted model of the cell membrane? Singer and Nicholson. And now let's see the function. What is the function of cell membrane? It is selective permeable membrane. Means it will select the thing which need to come inside and which need to go outside. This is the main function of the cell membrane, right? So please, at least what we are discussing, focus on that. Many more things are there, but meanwhile this is the small thing we are going to pick for the eukaryotic cell. So this is common in both plant cell and animal cell. Now next, if I am talking about the cell wall, this is a unique feature of plant cell, and can I say even in case of fungus you will find it, but in fungus it is made up of chitin. In plant cell it is made up of, like if I am taking higher plants, it is made up of hemicellulose, cellulose, and even of the pectin. Now among the plants, if I am taking in the case of alga, right, or algae, it is made up of which type of substance? Cellulose, galactans, mannans, and the minerals like calcium carbonate. Fine. This is the composition. Now what is the role of this cell wall? Can I say cell wall important function is to provide the shape to the cell. Fine, it is providing the shape to the cell. Even it is helping to, you can say, to protect the cell from infection, like any bacterial or viral infection somewhat can be stopped by this cell wall. Even it helps the cell from the protection against mechanical injuries. Right? Next, even it is acting as a barrier. Barrier against what thing? What thing is not required and big in size. That against that it is acting as a barrier. Fine. Now after this, even this cell membrane is maintaining no doubt shape, helping in protection, acting as a barrier. But when you will find, if you are keeping the cell into the isotonic, I mean say hypertonic or hypotonic solution, it will not allow the cell to burst or to collapse. Can I say it is acting as a support system to the cell also, so that it should not be burst or it should not be shrink if we are keeping into hypertonic and hypotonic solution. In hypotonic the cell can burst, in hypertonic it can shrink. So all that are the function of the cell wall. Now coming to the next point, cell wall I discussed, it is present in case of plant cell. Now let's see what is inside the cell. Inside the cell we are having this endoplasmic reticulum. But to start with this endoplasmic reticulum, let me to say you one point. There is endomembrane system. This endomembrane system will include such type of cell organelle which is showing the coordination among themselves functionally. Right? Functionally they are showing coordination among themselves. If they are showing functional coordination, they will come under endomembrane system. What will include under endomembrane system? It will include endoplasmic reticulum, right? Even it will include the Golgi bodies or Golgi apparatus. Even it will include their lysosomes, and even it will include the vacuoles. But all these structures having their distinct shape, size and structure over there, function even also, but they are coordinating with each other. That's why they are coming under endomembrane system. Now let's talk about each that one by one in short, so that important highlights we have to cover. Let's take for endoplasmic reticulum. For the endoplasmic reticulum, you must remember that these are the extensive , tiny tubular structures which are present around the nucleus or slightly away from the nucleus. Around the nucleus, if I go with the types, it will be the rough endoplasmic reticulum. Slightly away from that will be the smooth endoplasmic reticulum. RER stands for rough endoplasmic reticulum. SCR stands for smooth endoplasmic reticulum. Now let's check why its surface is rough. Because it is having on its surface ATS ribosomes. But in this case, ATS ribosomes are absent. If ATS ribosomes are present, can I say it help in the synthesis as well as the secretion of proteins? But this part, smooth endoplasmic reticulum, help in the synthesis of lipids and even of steroidal hormones in case of animal cells. Fine. So these are major difference between RER and SCR. Either it is SCR or RER. Commonly they are dividing the full cytoplasm into two compartments. Now what are that two compartments? Let's check on this point. One is the luminal compartment. Second is the extraluminal compartment. Right? Luminal compartment means, suppose I am drawing this as a lumen of the endoplasmic reticulum connected with each other. This part which is enclosed by endoplasmic reticulum, this will be luminal compartment, means which is a part of endoplasmic reticulum. Outside to that, this one, obviously this is in cytoplasm. So this part is indicating the extraluminal compartment. So this is the type of thing you should remember for endoplasmic reticulum. If you see the diagram of your NCERT, you will find RER is more close to the nucleus and smooth endoplasmic reticulum will be slightly away. They have shown you here and in the form of tubules. Because endoplasmic reticulum will have cisternae, tubule and vesicles also here. But tubular structure will be more in smooth endoplasmic reticulum. That's why here in this case they have shown the smooth endoplasmic reticulum with the tubular structure here. But the laminar cisternae structure more are in case of rough endoplasmic reticulum. They have shown you here, right? So this is about one of the member in endomembrane system, that is endoplasmic reticulum. Now let's check on the next organelle, that will be the Golgi apparatus. Do remember, Golgi apparatus was first discovered by Camillo Golgi. And this is which type of structure? This is a dense reticular structure which is present in the cytoplasm. If the word is given like tiny tubular reticular structures, then answer will be endoplasmic reticulum. If dense, dense why? Because they are densely stained. They are densely stained reticular structure. Even the endoplasmic reticulum are reticular structure. But here as per your NCERT, they are densely stained reticular structure. Even here there is a presence of cisternae, tubule and the vesicles, right? Now do remember next thing, what is the function of this Golgi bodies? For this one thing you should know, it is showing the polarity. Polarity means their cisternae will bulge like this and membrane to membrane like a flattened sac. They are piled on each other like concentric ring. Like one ring, its small part, one ring, their small part, like concentric ring near to the nucleus. Now as per the polarity, this is very important question. Don't miss to listen that. This convex side, this is known as a convex cis side or the cis phase, which you even call it as forming phase. Opposite to that, can you see it is somewhat bulged inside? That you call it as a concave trans phase. which you even call it as maturing phase. Now, if I will describe its function, as per their function, do remember this part of this Golgi bodies, like if I will take the nucleus here of a cell, this is a nucleus, you will find this convex side is facing towards the nucleus, whereas the concave side is facing towards the plasma membrane. This is a polarity. I repeat once again, this convex side is facing towards the nucleus and concave side is facing towards the plasma membrane. Now, why it is facing towards the nucleus? You know that around the nucleus, what is there? Rough endoplasmic reticulum, which is sending the vesicles containing proteins. It will fuse on the surface of convex, release the material into its lumen, and these all are linked with each other. So they will keep on transferring from lumen to lumen. Modification will happen. If protein is there, they need the foldings, whether it is tertiary, quaternary, based on that modifications will happen, and at last they will pinch out in the form of vesicle after getting proper maturation, and it is parceled. After packaging, it is parceled throughout the cell or outside the cell. This is the function of Golgi bodies, especially taking the things given by ER, then if required, then storing for short time, modification from layer to layer. When it gets mature, release through which side? Concave side it is going to release. That's why you call it as maturing phase. Fine. So this is important question based on Golgi bodies. Fine. Now let's take the next one. What is the next organelle? I am taking first of all for endomembrane system. Endomembrane system, first we discussed the endoplasmic reticulum, second we discussed Golgi bodies. Now next we are going to take about the lysosomes. Lysosomes you will find like a vessel, single membrane bound structure. It is filled by what thing? Various type of hydrolytic enzymes, which you commonly call it as hydrolases. Almost all type of hydrolases are there. For lipid they are having lipases. For the carbohydrate they have carbohydrases. For the nucleic acid they are having nucleases. For the protein they are having proteases. So all the type of hydrolases will be there. But condition is that will be working in pH acidic medium here. Acidic medium. Right? So whenever the cell organelle is not functional, lysosome is going to cover that and digest it. Whatever is useful it gives to the cell, what is residue it will throw it out. This is a function of lysosome. But once the cell is totally non-functional, then it will burst. Then you call it as what? Suicidal bags of the cell. Actually these lysosomes are packed by Golgi bodies. Right? So Golgi bodies are packaging that. They are packaging that and sending it throughout the cell. Right? That I already said you, that is a function of Golgi body, packaging and dispatching. So lysosome is even one of the result of it. Fine. So this is the next organelle under endomembrane system, that is a lysosome. Next after that, let me to take, that is a vacuoles. Vacuoles are even single membrane bound structure. This is even single membrane bound, right? But if I am discussing in case of plant cell, just now I discussed with you in the beginning of session, it is having single large vacuole, which is big in size, and whatever extra ions are there, they are picking it up. Like suppose this is plant cell and here is single large vacuole, outside is the cell wall. Now whatever extra ions will come, it will try to come inside the vacuole, but already ion concentration is rich inside the vacuole. So things are moving from low concentration to higher concentration by active process. So what is happening through that? Active process will happen through a membrane which you call it as tonoplast, right? And inside, what is present over there? Tonoplasm. And in the plant cell, approximately it is covering 90% of space is covered by single large vacuole in mature cell, right? Even in amoeba, another type of vacuole is there which you call it as contractile vacuole. This is in the case of amoeba. So whatever extra water comes inside the cell, it will throw out, like our kidneys are doing. So they are acting as osmoregulatory organ, or even you can call it as like part for excretion of the substances, right? Now next one more vacuole is there which you call it as food vacuole. Food vacuole again in the protist. Which protist I am talking here right now about? Amoeba again, or paramecium also. They are taking up the food and is giving it inside the body in the form of vacuole. Then that vacuole you call it as food vacuole, right? So these are the four structures under the endomembrane system. One we have discussed about the endoplasmic reticulum, then we have discussed about Golgi bodies, then we discussed lysosome, then last we discussed about the vacuoles. Now let's take the next organelle, mitochondria and chloroplast. I am taking that two structures in comparison. Reason why, the main we have to cover all the point in our this small session. So it's good that if we will compare and get the points out. Separate thing if I want to write, I will take you on the separate slide for it, right? So right now in front of you two membrane bound structure are there. Similarity between both is that both are double membrane bound cell structure. How many membranes are there? Two. So this is double membrane bound, right? But in case of mitochondria, the main function of this is that it helps in ATP synthesis. That's why you call it as powerhouse of the cell. But here the chloroplast, the function is the photosynthesis. Photosynthesis. That's why you can call it as kitchen of the plant cell. Kitchen of the plant cell. Now, next of the thing which are similar. See which are similar, I have written on the line itself. Both are double membrane bound. Both are semi-autonomous cell organelle. Semi-autonomous means they can perform their half function by their own. That is semi-autonomous cell organelle. Why they can perform their half of the function? Because both are having DNA and machinery like 70S ribosomes, and even they are having proteins, and both are having RNA in their matrix. But difference lies between that. In the matrix of chloroplast, you will find starch, but there is no starch in case of the matrix of this mitochondria. So can I say the mitochondria main function is the aerobic respiration, right? All the steps of aerobic respiration will happen in the mitochondria, like link reaction in the matrix, even the Krebs cycle in the matrix, and etc. will happen in inner membrane of mitochondria. Fine. Now let's talk about their individual structure. Individual structure for mitochondria, you will find this type of shape in your NCERT book. What type of shape it is? Sausage shape, right? Otherwise it can be cylindrical in shape also, and it is double membrane bound. But do remember, outer membrane is continuous, inner membrane is forming infoldings, which you even call it as a cristae. Do remember, this is cristae, not cisternae, right? And there is a matrix here. and I said you DNA is there, 70S ribosome, even proteins, RNA is there. And this two membrane, outer membrane and inner membrane, separate the thing into two aqueous compartment. One I said you matrix, this is intermembrane space, which is covered by this outer membrane and inner membrane. I repeat once again, this is outer membrane, this is inner, but inner membrane is showing the infoldings, which you call it as cristae. But outer membrane is what? Continuous, and these two membranes divide the mitochondria into two compartments, one into the matrix and next is into the intermembrane space. And function, just now I said you that they are working as the powerhouse of the cell, being they are forming ATP. Fine, and they will divide with the process of the fission. They will just divide and will duplicate itself. Now next is for chloroplast. Again it is double membrane. It is usually in higher plant disc shape. Otherwise their shape even changes from species to species. Various other shapes are even there. But as a structure, both the layers are continuous here. Right, there is no infolding, but sac-like structures are there, which you call it as thylakoids. And thylakoids are sac which are piled upon each other, and that one pile you call it as one granum. Such type many grana are there, which are further linked by stromal lamellae. Stromal lamellae. Now this grana and stromal lamellae will have pigments inside that. That's why they are helping in light reactions. Whereas the matrix part, this matrix you call it as stroma. It will show which type of reaction? It will show the dark reactions, which we will discuss in the photosynthesis chapter. Means all enzymatic actions will happen in stroma part. And additionally, I already said you DNA will be there, even it is having 70S ribosome. They can manufacture their proteins so that half of their function they can perform independently. Fine. Okay, so this is about important structure chloroplast in case of plants. Otherwise plastids there, you know that it is of three different types. One is chloroplast, chromoplast, leucoplast. Being chloroplast is more important to discuss as per your NCERT book linked to the photosynthesis chapter. We have taken their structure. Now coming to the next, we are having the ribosome. Just now I said you which type of ribosomes. In eukaryote it can be there, it can be 80S, it can be 70S. Can I say they are helping in the formation of the proteins? 80S, where it is present, I mentioned in the last lecture also, it is in eukaryotes in the cytoplasm part. Even it is present on RER, even it is present on outer surface of or outer membrane of nucleus. Whereas 70S will be present, just now I discussed, in mitochondria, and even it is present in case of the chloroplast. And what they are doing? They help in the formation of proteins and they are membraneless structure. And all either 80 or 70, they have two units, subunits. One is a larger subunit and one is a smaller subunit. In both the cases, smaller and larger are there. Their sedimentation will be different in both the cases, right? So this is membraneless structure both in eukaryotes as well as in prokaryotes. Fine. Okay, so let me now take these two structure in short here, and it's very important, we cannot miss that flagella and cilia both are for locomotion in case of eukaryotes, right? But if we cut it transverse section, you will find inside it is covered with the help of, first of all, plasma membrane. The green color line is a plasma membrane, and both are showing 9+2 arrangement of microtubules, right? 9 means they are having the 9 doublets, right? And 2 means they are having 2 singlets. 9 doublets, 2 singlets of what thing? Microtubules. And both are helping in locomotion. Rest of the thing, singlets are covered by this, you can say by this sheet, central sheet. And these doublets, which are 9 toward periphery, linked with this central sheet with the help of radial spokes. And doublets are linked with each other with the help of this linkers. They are helped, or you can say inter-doublet bridge. Inter-doublet bridge. Fine. And singlets are again linked to each other with the help of bridge. This complete exoneme which is inside the plasma membrane, all is proteinaceous here, both in cilia and flagella. Cilia will be small in size, big in number. Flagella will be long in size, length is more, but number will be less. But internal structure will be 9+2 arrangement of microtubules. Fine. Function is what? Locomotion. And they are the outgrowth of cell membrane. See, this point is very important. In prokaryote, flagella is there, but that is cell wall extension. But in eukaryotes, these are the cell membrane extensions, right? Now, next after this is for centriole. I said you even these are membraneless, and this is present in animal cell, few of the lower plants. But they will show 9+0 arrangement. What 9 here? 9 will be the triplets now, which is towards the periphery. And in the center there is no microtubule. Only central hub is there, which is proteinaceous. Proteinaceous hub is there in the center. Periphery, this is one triplet. How many triplets are there towards periphery? 9 triplets are there. Means 9 into 3, 27 microtubules are there in one centriole. And this structure, you call it as cartwheel-like structure. You call it as cartwheel-like structure. And what is the function of centriole? It helps in the formation of the spindle fibers during the spindle formation. And even if suppose this is flagella or cilia, their filament part, the basal body will be created by this centriole. Means basal body of cilia and flagella is having 9+0, but their filament part is having 9+2 arrangement. And can you see there is no membrane around it? That's why it is membraneless. And all triplets are linked by the linkers again, right? But there is no singlet in the center. Radial spokes are there, both in case of centriole transverse section and even in case of this cilia and flagella transverse section, right? So do remember at least these major points for the centrioles. Now last organelle which we have to take, that is the nucleus, boss of the cell, which is controlling all the activities on the cell, controller of the cell. The main important point, the stuff which is present inside that you call it as what? That is the nucleoplasm. And this nucleoplasm is surrounded by nuclear membrane. That's why this is a true type of nucleus. And chromatin material is present here in the nucleoplasm. And this chromatin material consists of double-stranded DNA, right? And which is linear and having histone proteins, right? Otherwise this chromatin material, not only DNA, even it is having small amount of RNA, because transcription happen in the nucleus. And along with histone proteins, even they are having non-histone protein also. Now why the word chromatin? Because it will pick up the stain. That's why you call it as chromatin fibers, which are going to be extensive, spread throughout the nucleus. Elaborate means complex. Even it is going to be indistinct, being it is thread shaped, tangled with each other. That stage is found in which? Interphase. This type of nucleus you will find in interphase. If I am discussing for human diploid cell, in human, how much material of chromatin it is having? Approximately 2.2 meter for 46 chromosomes it is having, right? Now the structure which is present inside that, one more important, nucleolus. Do remember, nucleolus is what? Membraneless structure. It is not cell organelle. This is a structure which is present inside the nucleus and help in the formation of rRNA, which will further help in the formation of ribosomes, right? And very important question which is frequently asked in the NEET is that nuclear membrane is double membrane. That's fine. Outer membrane, inner membrane. But outer membrane is having the ribosomes on it, which you call it as ATS ribosomes. And this outer membrane is continuous with which type of cell organelle? That is the rough endoplasmic reticulum. Fine. And between the outer membrane and inner membrane, there is a perinuclear space. What is present? Perinuclear space whose dimension is 10 to 50 nanometer. This is very important question. In the match the column they had asked. Now this chromatin material during cell cycle cell division can form the chromosomes, and that all we will take further if session is there, then in cell cycle cell division, right? So these are the few things important we have covered in a eukaryotic cell. Please do read NCERT, stick to that with the diagrams also, and just watch this video. If any queries are there, then we can take it. Thank you very much, class.